Look at the categories that the different amino acids fall into, and see if we can understand why. The more we understand these different types of R groups, the better we can understand the folding that takes place with an amino acid. This is going to become really important as we try to understand how a protein functions. And then we're going to look at peptide bond formation specifically. So how two amino acids are linked together through a dehydration synthesis reaction. So the first and largest group of the 20 amino acids are the nonpolar. And what we mean by nonpolar is that the R groups have no polarity or charge associated with them. So you can see glycine here with just a simple proton. Alanine here with a methyl group. We can go on down the line. Valine is all hydrocarbon. Leucine, the R group, is all hydrocarbon. Isoleucine, the R group, is all hydrocarbon. Methionine, which is always the first amino acid in the sequence that's put into place as a new polypeptide is formed, it's mostly hydrocarbon, but it has a sulfur in there. Now, sulfur acts a lot like oxygen and nitrogen in that it's an electron hog, right? It's highly electronegative, and it's going to draw electrons towards it. In particular, when it's an SH bond, that will be a highly polar bond, just like an NH or an OH bond. However, when sulfur is just sandwiched between two carbons, those bonds are not particularly polar. So this would still be a non-polar R group that we see down here in methionine. Now we move over here to phenylalanine. We've got a methylene group and then a benzene ring. Remember that in shorthand for organic chemistry, each point in a structure like this represents a carbon. The two lines represents a double bond. And even though hydrogens aren't drawn on here, they are implied. So phenylalanine, all hydrocarbon. Tryptophan, all hydrocarbon with the exception of this NH. And we know this NH is going to be a polar bond. But if you look at the size of that R group, that tiny little polar bond, one bond amidst all those carbons and hydrogens, is not going to be able to lend any significant polarity overall to that R group, and water is not going to play very nicely with it. And then finally, proline we see is also all hydrocarbon. Now, one thing you may have noticed if you're looking at this is the way they've drawn it. They've drawn the amino group as if it's acidic, and they've drawn the carboxyl group as if it's alkaline. That is just one common convention. So you can draw them like this. If you do, be sure you include the symbols for the charges. NH3 has to be positively charged. COO has to be negatively charged. So if you choose to draw them in their altered states, you feel free to do that, but make sure you include those charges with them. Otherwise, you can just draw them neutral, where you've got NH2 on one side and COOH on the other, and everybody knows exactly what you're talking about. So the first group of amino acids, which contains 1, 2, 3, 4, 5, 6, 7, 8, 9 of the 20 amino acids. These are the nonpolar amino acids. These are going to hide from water. They're going to be embedded in membranes, embedded in the interior of a folded protein, etc. The other two groups are the polar and the charged groups, and hopefully you can see why they're called polar and charged groups. So here we've got serine, has an OH, polar. Now, unlike the one we saw on the last slide that had an NH, this is a pretty small R group. And so a single polar covalent bond on a pretty small R group is likely to lend some polarity associated with it. Same here with threonine. Here's cysteine. Remember I said that SH is a polar bond. And so here's a significant polarity. Here's tyrosine with an OH down here. Asparagine has this odd arrangement where it has a carbonyl carbon and then an amino group. Glutamine has the same functional group here. These are all polar and are all going to want to be interacting with water as the protein folds. Now, the charged groups can be considered acidic if they have a carboxyl group, and therefore negatively charged under neutral to alkaline conditions. Or they can be called basic or alkaline when they have one of these amino groups that can pick up a proton, and they'd be positively charged under acidic conditions. So you can see aspartic acid and glutamic acid. Then we've got lysine, arginine, and histidine that all have these charges associated with them because of the amino groups. These guys will also want to interact with water, but they may also want to interact with each other because there's a potential for ionic bonding between the R groups, and that can become very important. Now, what we want to do is we want to ask, well, how do these guys come together? How do individual amino acids link up to polymerize to form a polypeptide? In the purple box you see here, you can see you can recognize the backbone of every amino acid. You've got an amino group, you've got your central carbon coming off of the hydrogen, and you've got what was your carboxyl group. Okay, your carboxyl group then is going to enter into a covalent bond with the amino group of the next amino acid. So you go from a carbonyl carbon, which was once a carboxyl carbon, to an amino nitrogen. That bond is called a peptide bond, and it's what links the amino acids together. And here we've got the R groups. So we end up with a backbone that is, we can call it the peptide backbone, where it's all identical. Right? Everything looks the same going down the line. And what's different is at each position which R group is waving out in the breeze and available for chemical reactions either with water or with other R groups or with substrates in an enzyme or whatever it happens to be. Now you notice it's a dehydration synthesis reaction like we see here. The OH of the tail end, and we call this the carboxyl end of our growing polypeptide, is removed, as is one of the hydrogens of our amino group, and it creates water, leaving behind our peptide bond that you can see down here. The very first amino acid that's put into place has an unreacted amino group, and we call this the amino or N terminus of the chain. You continue on down, in this case only 3 amino acids later, but in a typical protein, a few hundred amino acids later, you come to the C or carboxyl terminus, where you have a carboxyl group that is now unreacted because there's not yet another amino acid coming in. Some pretty remarkable diversity can be created. If you've got even a short polypeptide with just 100 amino acids in it, you can see down here on the bottom the math, 20 possibilities in every one of those 100 positions. You've got about 10 to the 130th power possibilities of stringing them together to make different proteins. That's a 1 with 130 zeros behind it. It's a number we can't really even wrap our brains around. So much like the nucleic acids, the incredible potential diversity among polypeptides and thus proteins because of the nature of how they polymerize and how long they are and the diversity of amino acids is practically limitless. Pretty remarkable. So let me suggest you go back through this series of slides, take a look at the polar, the nonpolar, and the charged amino acids. Make sure you can recognize them and group them based on those R groups that you see. Make sure you understand the rationale behind them. And then take a look carefully at this polymerization slide here so that you can see the dehydration that takes place. And you can see the backbone that's forming with the R groups waving out in the breeze, as I like to say. All right, let's go ahead and shut this one down.